This video was made by Team CRISPR for the Northwest Association of Biomedical Research Student Expo 2017. None of the images represented in this video are the property of this team. What is CRISPR and how does it work? The human body, despite all of its capabilities, still manages to be rather limited. We suffer from countless diseases, ailments, and disabilities. The most athletic of our members can only run at about 20 miles per hour at maximum. We have horrible night vision compared to some birds and many other species. Our smelling and hearing are many times worse than a dog's, and our genetics are also flawed in many ways that open us up to diseases. Genetics have given us superior intellect, which has separated humanity from animals. While other species have their survival advantages and disadvantages, just imagine if humans could have all of the preferable traits without sacrificing any of the strengths. Now, with this revolutionary technology of CRISPR, we can enhance our natural abilities and fix our genetic issues in ways we never thought were possible. There's a lot of controversy surrounding CRISPR as it falls under the umbrella of genetic engineering. While ancient people practiced selective breeding, they were modifying the genetics of plants and animals. In this way, we have been manipulating the genetics of organisms since the dawn of agriculture around 12,000 BC. In recent history, genetic engineering has been mainly used in edible crops such as corn and wheat. Scientists have been manipulating genes of these plants to make them grow bigger, more nutritious, more pest resistant, and to give them the ability to grow in different climates. The science shows that GMOs have a longer shelf life, are more nutritious, and are less likely to be infected, but also pose an allergy risk and can lessen the effects of some antibiotics. This sparks an ethical debate. Many people claim that it's morally wrong and playing God. With the discovery of CRISPR, people are finally starting to practice genetic modification, while this allows them to focus on the traits that they want to change rather than just hoping for the desirable changes like what farmers did with crops. Now let's talk about CRISPR's technique of genetic engineering. We'll start with viruses. Viruses inject their RNA into a cell bacterium. They accomplish this task by making a protein key that fits into, you guessed it, a protein gate on the cell, opening the cell along the transpoint of viral RNA through the cell membrane. The virus hijacks the protein synthesis process inside the cell to make inordinate amounts of that virus's RNA. Eventually, the cell ruptures and the viral RNA is released into the host, free to infect more cells and repeat the process. To overcome this infection, the host immune cell needs to carry and destroy the cells infected by the virus. So what does CRISPR do? First of all, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR is also a great big molecule that captures viral RNA and places it in a long string of all the previously captured RNA. These segments of viral RNA are separated by repeats. Repeats are basically the genetic white noise which helps them to separate and identify one from another. Then, a protein called Cas9 takes the virus RNA from CRISPR and compares it to the human being's own RNA to check if the person has been infected with that virus. If the RNA of the virus is discovered inside the RNA of the human being, then CRISPR-Cas9 molecule will remove the viral RNA from the cell. That is how CRISPR-Cas9 can remove the infection from the human. So how can the magic of CRISPR-Cas9 be manipulated to perform medical miracles? Scientists can inject the patient with CRISPR molecules that contain RNA for a defective genetic trait in the patient. Cas9 then proceeds to hunt and remove the selected defective trait from the cell in the patient. This process can also be used to add traits. So in summary, the process by which CRISPR-Cas9 can edit out viral RNA from a cell, the CRISPR-Cas9 molecule can also edit traits from the human cells. Genome editing through CRISPR has, in the short period that it's existed, already revolutionized many areas in the medical field. A few of many items on CRISPR's accomplishments is that, in a work published by the Proceeding National Academy of Sciences, researchers made a mosquito malaria resistant by using CRISPR-Cas9. That, along with what's called a gene drive, made 99% of their spawn malaria resistant too. This could possibly cure malaria completely in certain locations, saving millions of lives. In Xuan University of Chengdu, China, scientists injected a person with cells modified by CRISPR-Cas9. The trial involved modifying a patient's own immune system to make them more effective at combating cancer cells and then injecting these modified immune cells back into the patient. In the future, CRISPR may also be able to immunize humans from biological scourges and may also improve our biology in ways we never thought possible. To bring this presentation to a conclusion, we'd like to summarize a few main points about our research. Genetic modification has been going on ever since the dawn of agriculture, whereas CRISPR is a much more recent technology that we've only just started using to modify cells. Although we can't always predict where technology like this will take us, we humans should always be thankful to be alive during this time of rapid technological advancement, especially under the subject of biotechnology, where this new technology is so cheap and easy to use, almost anyone can do it. For example, in our own lab, we took a CRISPR kit to edit genome of E. coli bacteria to enable the bacteria to grow in streptomycin-laced growth medium. This required minimal effort. However, there will need to be regulation to make sure this type of genetic editing doesn't get out of hand. Thank you for listening to the description of our project. We are overjoyed that you are interested in CRISPR and the ways it can be used to help improve the health and productivity of our species. We would like to thank our mentors and the sponsors of the Northwest Association for Biomedical Research for inspiring the middle and high school kids like us to learn about topics such as CRISPR and inspiring us to think about the future of our species as a whole. We hope that CRISPR will be used to end genetic diseases and push human health forward into the 21st century.